This will be a review of Chapter 3, the chemistry of organic molecules and the 4 main biomolecules of life. And so in this chapter, you'll be responsible for understanding the chemical structure and function of carbohydrates, lipids, proteins, and nucleic acids. As you move through the chapter, it will discuss the carbon atom and how the carbon atom has 4 electrons in the valence shell, which allow it to bind with 4 other atoms or double or triple bond with a similar atom, but it needs 4 more atoms to be considered, for that outer shell to be considered full, or for that atom to be happy, we call it. We discuss how the carbon atom can form long chains of hydrocarbons, of carbon and hydrogen bonds that we can attach a functional group to change the function. The molecule could then go to do something else. It could become polar or nonpolar, soluble, non-soluble in water, but we can essentially recycle and reuse the basic components of our molecules. Our 4 main biomolecules, again, are carbohydrates, lipids, proteins, and nucleic acids. Organisms metabolize these molecules to either access or store energy. To build things up is called anabolism. To break things down is called catabolism. But in some way, we're either forming or removing portions of these molecules to access that energy source, or we can use them for structure, etc. The first are carbohydrates. So you'll need to understand that monosaccharide means 1 sugar, and these monosaccharides are used for an immediate energy source. An example would be glucose or fructose, a 6-carbon sugar. The nucleic acids, DNA and RNA, both contain a sugar, either ribose or deoxyribose, respectively. A disaccharide, that's just 2 monosaccharides stuck together with a bond. Again, the body uses these for quick energy. Examples would be sucrose or lactose. Sucrose can be seen as being a glucose and a fructose molecule stuck together. Lactose is glucose and galactose. So to break these down, the body would have to separate those to get to that glucose molecule or an isomer of glucose to use to essentially access those electrons, to access that energy source. To store energy in a long-term form, we can make polysaccharides, which are large chains of sugars linked together. Plants store energy in the form of starch. Animals, mammals, store energy in the form of glycogen in the liver or in the muscles. The cellulose in plant cell walls is also composed of carbohydrates. The chitin in fungus or exoskeleton of arthropods is a long carbohydrate. That takes us to lipids. Lipids are long chains of hydrogen and carbon. There are 2 main types. We call our lipids fats. That's sort of the colloquial term. There could be unsaturated fats, or saturated fats. Saturated fats are just a long chain of carbohydrates, or carbon with those hydrogens off to the side. We say they're saturated with single bonds. An unsaturated fat may have 1 or more double bonds, which makes it kink or change shape. And in biology, you change shape, you change the function. So your saturated fats are going to be solid at room temperature, while your unsaturated are going to be oils. Animals store fat in the form of triglycerides, which are these fatty acids with a glycerol group attached. We use lipids for long-term energy or insulation. Plants can't get fat, but they can store oils. That's how their triglycerides are stored. Again, these would be for long-term energy storage. One of the most important lipids we cover in this chapter is the phospholipid. It has a large polar phosphate head. That means the electrons are going to spend more time on this side of the molecule. And these nonpolar tails, a saturated fat and an unsaturated fat. The polar head is going to be hydrophilic, water-loving. The nonpolar tail, hydrophobic. That allows the cell to use these molecules to create this bilayer and create a separation from inside and outside of the cell. Another form of a lipid is a steroid, which is just a fused carbon ring. We can use steroids as hormonal messengers and as structure. And so cholesterol allows us to give our plasma membrane structure. Waxes are another form of lipid. They're used for protection. We don't make much wax, just ear wax. But if you're like a plant, you'd make a lot of wax to coat you in a cuticle to prevent water loss. But again, waxes are for protection. If carbohydrates are made up of glucose monomers, proteins are made up of amino acids, polymers of amino acids linked together by peptide bonds. Another name for a protein is a polypeptide. A protein is just a polypeptide that's been folded into a particular shape. And proteins do a lot of things. They're a major part of living organisms, but they aren't just for structure. We utilize them for metabolism. Enzymes are proteins. Support, transport, the hemoglobin in our blood transports oxygen and carbon dioxide. Our antibodies are proteins, so we use them for defense. We use them for hormone regulation. Some proteins act as hormones, and of course for motion, either in large muscles or microtubules inside of our cells. All of these could be a function of protein. Essentially, DNA is the blueprint for how we make a protein, and the protein is an expressed trait. So an amino acid is just a group, a carboxyl group and an amino group attached to a special variable or functional group. There are 9 essential amino acids that we have to eat, 11 that we can make, so we call them non-essential. Proteins can have 4 levels of structure: primary, secondary, tertiary, or quaternary. Primary is simply a long strand of amino acids. Secondary, we fold those strands into a particular 3D shape, most notably either an alpha helix or what we call beta pleated sheets. But they are coiled and stabilized because when they're folded, their atoms and electrons can interact and form hydrogen bonds, little static attractions. Tertiary proteins are your globular proteins. They've been balled together, so now there are more hydrogen bonds, more covalent ionic bonding. They're a bigger structure. And finally, the 4th level is the quaternary, or the superstructure. An example of this would be the hemoglobin, which is 4 polypeptide strands all molded to do one big job. So here's an example of a primary protein structure, secondary, tertiary, quaternary. The proteins come in a variety of shapes and sizes. They are essentially expressed traits. Nucleic acids are the final biomolecule we talk about in here. They're made up of polymers of nucleotides. Now, a nucleotide, maybe a new word, is just a special sugar, a 5-carbon sugar, a phosphate group, and a nitrogenous base. In DNA and RNA, these are nucleic acids that do special things in our cells. The sugar is what differs. In DNA, you have deoxyribose. In RNA, you have ribose, a phosphate group. And these sugars and phosphate groups make an alternating backbone of sugar, phosphate that allow the nitrogenous base to stick to that. And so we call that phosphate sugar a backbone or a basebone. Polymers are nucleotides. Here's an example of a nucleotide. So we have that pentose sugar, 5-carbon sugar, a phosphate group, and then a particular nitrogenous base. In DNA and RNA, the nitrogenous bases differ. You could have 4 different types, but these nucleotides form DNA or RNA. Another example of a nucleic acid is ATP, the energy currency of the cell. We can use nucleic acids for cell signaling in the form of CAMP, or as cofactors, which we'll talk about in Unit 2 as coenzyme A, FAD, NAD, or NADP. You'll need to know the difference between DNA and RNA. DNA has the sugar deoxyribose, RNA ribose. In DNA, we have 4 nitrogenous bases: adenine, cytosine, thymine, guanine, or A, T, C, and G. In RNA, we have adenine, cytosine, guanine, and uracil. So if we say uracil, we're talking about RNA, not DNA. DNA has thymine. DNA is the genetic information inside of our cells. RNA allows us to create proteins. It's the regulation of gene expression. They're not vocab words now, but they will be in the future. Pyrimidine and purine. We need to understand that pyrimidines, cytosine and thymine and uracil, are single-ringed nitrogenous bases, while purines, adenine and guanine, are double-ringed. So you may as well start learning them now. We'll talk about them again when we talk more in detail about DNA. Nucleic acids in the form of DNA, when those molecules are bound together, the single ring and double ringed molecules bind together in what we call complementary base pairing. So we say A is paired with T, C with G. Other nucleic acids include ATP, the energy currency of the cell, and coenzymes.